Okay, so if we can all be upstanding for the official party. And we're going to do this by celebrating it in a Professor Chris Pemberton way.
I think people... E ka mana, e ka reo, e ka mauka whakahi, e ka awawa, e ka pātaka o ka tauka tuku iho, tēnā koutou, tēnā koutou, tēnā koutou katoa. I think that's the first time I've been rickrolled at the university. For those who don't know me, my name's David Murdoch. I'm the Vice-Chancellor of the University of Otago, and I'm really glad I got through that last sentence because the temptation standing here is to say Dean and Head of Campus, so I got through that. Big welcome to everybody. This is a very special occasion, as Suze's indicated. This is the celebration of Chris Pemberton's promotion to Professor. And really this is one of my great pleasures, the great pleasures of my role, is to take part in these inaugural professorial lectures. This, as you're probably aware, we've put off a lot of them, been very disrupted by the COVID pandemic. We're now catching up. Chris is the fourth this week, and it's really been wonderful to be able to hear from our academics from all over the university. And now we have them live streamed and are recorded, and you can watch them afterwards. So I really encourage you, I know coming from health sciences, you tend to focus on health sciences, but I really encourage you to look around at the other IPLs that are happening at the moment and see what our amazing colleagues are doing around the university. My main role is to welcome everybody, and so big welcome to everyone who's here, everyone who's joining online, either live or delayed. And I know coming from the wider university community, but also the wider Christchurch community and beyond. But special acknowledgement to Chris's whānau and friends. And I think here today we have wife Megan and daughter Rebecca. Jacinta is here. I think Mike was not unable to join, or maybe joining later. And also I know there are quite a few other whānau and others who will be watching online. So a big welcome to everyone. It's really wonderful to have you here today to take part in this wonderful event. And before I hand over to Richard Geary, who's going to give more detail about Chris's background, really just a personal note, Chris, we've known each other a while, and I really have enjoyed all the interactions we've had over the years, enjoyed your enthusiasm and passion for your research, but also just really want to acknowledge that you are one of our staff and scientists who have a very good commercial instinct, and I'm sure you'll be reflecting on that in part of your talk, and that's something that university scientists don't often have. In fact, it's uncommon, and you certainly have had that instinct throughout, I guess, throughout your career, certainly the career that I've known. So congratulations. This is really a well-deserved promotion. Really looking forward to the lecture, and I will pass over to Richard Geary. Nō rea, tēnā koutou, tēnā koutou, tēnā koutou katoa.
Tēnā koutou katoa, ko Richard Geary toko ingoa. It is my absolute pleasure to introduce Chris Pemberton to you all today. Although if you're here, you probably don't need much of an introduction to him because you're here to support and to celebrate the amazing work that he's done over such a prolonged period of time. David just said that this is one of the best things he does in his job. This is the best thing I do in my job as head of department, because really how well the department does, its successes are reflected in its people. And today we have one of the great people in the department coming forward. And I've known Chris for a long time. I think we've been around the department probably for a similar time. And I think even I flattered with someone who was a research assistant with him way back at the very start of this journey. So really a lot of stuff has been going on over a long period of time. And he makes up one of the really important aspects of the Christchurch Heart Institute, which is a really strong group in our department and the school and the university as a whole, so it reflects that as well. So here's the biography to tell you all about him and what he's been up to. So Chris began his research at the University of Canterbury, where he completed a Master's in Animal Physiology, majoring in neurophysiology. He then spent 14 months at Lincoln University trying to coax deer into a research study holding pen. But he gave up and came to the University of Otago in Christchurch to do his PhD under the guidance of Professors Espiner and Yandel. After completing his PhD on the biochemical forms of BNP and experimental heart failure, he took his family to the farthest place away from New Zealand in the world. That was to Finland, where he gained postdoctoral training in isolated organ techniques and making angels in the snow to celebrate PhD celebrations. So we can't do that today. I'm not quite sure if angels in the dirt really work quite as well as in the snow. Upon his return to the Department of Medicine at University of Otago in Christchurch, he established the Isolated Heart Facility at the Christchurch Heart Institute, led by Professor Mark Richards. And using this facility both as a research discovery tool and physiological testing platform, the main focus of Chris's research has been the discovery of circulating factors in the blood that could potentially be used to address areas of unmet clinical need in the diagnosis and management of patients with cardiovascular disease. In 2011, he was awarded the Health Research Council Lilly Medal. And I just wanted to pause there because the Lilly Medal is one of the most amazing awards you can get from the HRC for an amazing breakthrough. This is not just a career of research, but one piece of something, a breakthrough. So for Chris to get that from our school here, I think is an amazing achievement. I'd just like to really acknowledge that here. And he currently heads the Translational Biodiscovery Laboratory and is a member of the senior leadership team of the Christchurch Heart Institute. He's also Chief Scientific Officer of the diagnostic company Upstream Medical Technologies, which he co-founded in 2015. So Chris's talk today will cover a wide range of study areas, including going right back to his master's, and will include caterpillars, an attempt to rewrite a small part of textbooks, how to tell if someone has a dodgy coronary arteries, and a bit of a side hustle involving COVID. Chris, thank you very much. You're very welcome to come up here and tell us about your amazing career to date. Thank you.
Can you hear me? That's a shame. So we have prestigious Maori Dean. It's Maori Language Week, and apparently I'm the first person to wear this cloak. No pressure. Ka mihi ki te kairahi o te whare wangana o Otaku, VC Prof Murdoch, Prof Peterma, Prof Priest, Prof Geary. Oku hoa mahi mai I te CHI mi te UOC whanau. Ka mihi ki te toko me te noho mai o taku whahine, whanau me aku a hoa a ringa ipurangi, koto ta toko ti takianga, o nea mea katoa e ruto I nā tau ka haere tonu. E mihio ana hau ki nei tuahauriri te mana whenua o tēnei rohi. Ki mihi ki o koto kitia kitanga o te pai o te tahi. Ko mihi hoki a ohu ki te whenua o ko tapuna no ingarangi. Kai hairanga o ko tapuna ki Aotearoa e tupu I rei o e ringi I te kokore o Iraki. Ki mahi tahi te tau he pai no mā tangata katoa ki ko tahi I arau I te rangatira o Iha Kariti. Ka mutu, ka mihi I ka tauhunga katoa I mate e tēnei rei, ke pai tino karero mo tō wama te aro. Tēnā koto, tēnā koto, tēnā koto. Au katoa. Apologies. That was awful. Thank you for turning up. I know you're here for the food, but you've got to listen to me for the next 30 to 40 minutes. So I would just like to say, it's nothing personal, but I really enjoyed rolling you all. That was something I wanted to do for a while. So I'm going to talk about what I've done and the people who are most important in the time that I've spent here at this fantastic institution. So the first biology lesson that I got was that trout are worth catching. I got that from a very young age. The second one I got was that dogs are great mates and will drink beer, but they're absolutely useless at digging ditches. Going to school, well, that was not very good, and the only person who really understood me was my French teacher, who wrote Chris would do a lot better if he stopped chatting in class. And I think that's quite a common thread among a lot of students at that time. And we all start out with bright ideas and a fresh new bag and looking good, and then somewhere along the way it all turns to custard and you end up looking like that. That was at the end of year swimming sports at school. Going to university, that was a lot of fun, and we had a great group of people in my master's cohort, really smart, intelligent, fantastic people, and as per usual it all turned to custard on Saturday night. And what's really scary is that this guy here was learning how to fly Skyhawks for the New Zealand Air Force, and he's probably taken a lot of people around the country on a 737 or something now, so that haunts me still to this day. So caterpillars, that's the first part of it, and as a bright-eyed student, the project that I undertook in neurophysiology was to figure out what the piranha moth larvae don't like to eat. And the piranha moth are those really big moths that fly around in the spring. They're really annoying. Birds love them because they're very tasty. But their larvae are actually quite a big problem, and they were a big problem at the time. It was worth tens of millions of dollars of lost agriculture because they would get into the grass and they would literally, you could go into a paddock and you could lift it like carpet. They would just ruin everything. But the best things about it that really intrigued me was that you got a free Apple computer from the DSIR, and there was $6,000 of working expenses. And that's them there, these little critters that live in the soil, and they basically eat away like the grass grub and other things as well. And they've got a face only a mother could love. And that's the scanning electron micrograph of what they look like, because the first thing we had to do was figure out, well, how do these things sense what's around them to know what to eat? So we took a lot of pictures on the SEM mugshots like this, and it's these things here. So this is at the end of its antenna, and this is on its mouth around here. And these things are actually sensory organs. So there's quite a few of different lengths on the antenna, and there's quite a number of different arrays at the on the mandible and in the maxilla as well. And the way that they're structured is that they have a hole at the top, or a pore, and you've got these axonal projections which run along here down to the axons, and they taste literally the whatever's in, and you can have these as a fluid sensory or an air sensory system as well. And you can visualise these with a stain called methylene blue, and it's probably not as apparent, but you can see the blue axons here and they extend up to there. So it's a selfie of a bug tooth. And then the primary reason, once we'd done all this anatomical assessments, the next thing we wanted to do was, well, what do they like to eat? Can we figure out what they don't like to eat? And to do that, you had to record it using all these electrodes. So you had a glass-filled pipette, and it had a sodium chloride and silver chloride electrode in it, and you could change whatever you wanted in the fluid in that electrode, and you put it over the maxilla, and you could measure whatever you wanted. And it was in this setup here, and you could have the recording side, so you basically cut the head off the caterpillar and put it in a bath of sucrose, and that would keep it alive for a couple of hours. And then you put it all through an amp, and it would go through another quad amp to an oscilloscope, and you could record it and put it onto the amazing Apple computer that we had, and you could also put it to the speaker. And there was a PhD student who was looking at another insect at this time in the lab, and we figured out that we could reroute the speaker to play whatever we wanted. You could dampen down the signal from your experiment and play music through it. And when the supervisor was actually there, of course, we had to play something that was, suitable. He was okay with that. But when he wasn't there, it was more like that. That was a lot of fun. And so eventually, by the end of it, we'd worked out the structure of what the caterpillar's sensory organs were like. And then we thought, okay, what are we going to put into them to see what happens? And the very clever people at the DSIR who were funding the project had worked out that there's this thing called phasolin, which is quite common and it's quite abundant in legumes and beans and things like that. And the caterpillars don't eat the beans. And they worked out what the structure of this thing was. And for the chemist, it's a pterocarpin derivative isoflavonoids. To the non-chemist, it's just a thing with a whole lot of rings on it. And you could get a different structure. It tastes awful. I'm not going to say how we found that out, but it tastes awful. And what happened was that we would put this in the solution with a whole bunch of different things. And the caterpillars, they really loved sucrose and fructose, but their big thing that they loved was phenylalanine. And if you put 0.1 mole of phenylalanine on and you had the recording, you can see all the impulses going off crazy like here. If you add the phaseolin, disappeared, it was gone. And we thought, this is pretty cool, we found something they don't like. So then the whole idea was you could naturally breed it or genetically introduce phaseolin into the grass, and you would perhaps reduce their preference for feeding it. And this was my first introduction to how bureaucracy can affect science, because then at that point the DSI completely restructured. All that work was lost and it went off into something else. And so that was the first lesson learnt. And this is a book I used to read as a kid about a schoolboy called Nigel Molesworth. And a rule of life, there's only one thing in cricket, and that is the straight bat. Keep your bat straight and all will be right in life as in cricket. But when my bat is straight, I still get bowled. Is that an omen? So have a slosh. You're going to get bowled, but it's much more satisfactory to go out in a blaze of glory. So at that point, you have a master's and things are looking pretty good, and you think this is quite good, I want to carry on. But I got distracted. Didn't I? Yes. And I went off to Lincoln, as a lot of people here have done. The number of people actually here at the medical school who in some phase or form have gone through Lincoln is incredible. I went to work for Prof. Graham Burrell looking at deer antlers. And the whole interesting thing about them is that deer grow antlers in the summer and then they run around, but then they take all of that bone growth and ossification, everything, and they put it back into their skeleton. So the whole point of that project was, if we can figure out how they do this, then there may be therapeutic treatment options available for osteoporosis or things like that. And so the structure of the deer antler, they have all these different cells. You've got, I can't see here, I'm going to put these on. This is what happens when you become a professor, you need glasses. They've got these reserve mesenchymal stem cells here at the top, at the end of the antler, and you've got all these other different cell types. And so we were sampling from the neck veins to try and get through a representative sample of what was going on inside these antlers. Now, that only lasted for about 12 months because while having the deer in the crates like that photo shows, you only use hinds because they're much more pliable, and you get in there with a stag and it's all over. And you had to come from behind the stag and the hind and put your hand over the eyes like that, and of course I was the guy who had to do this. And that was usually the result. And so after several repeated statements from deer and some fairly highly acute inflammatory responses from you-know-where, I thought, no, I've had enough of this. And ended up coming to Christchurch. And at that time, well around the circles, the reputation of Prof Espiner and Tim Yandel as real gung-ho, this is the team you want to work for, that was apparent. So I came in and I presented myself, and they must have seen something useful, and they agreed to take me on as a PhD student, for which I'm eternally grateful. So the question at that time was BNP. Now, the Christchurch Heart Institute talks a lot about BNP, we know a lot about it. And the problem that they had at the time was that it was emerging as a potential cardiac marker, and it's been a marker now for a number of decades now. And it's released from the heart in response to increased wall pressures and stress, and hypoxia as well can make it release. And it does this, and there was a big problem at the time. Patients with heart failure, they would come in and they would have breathless symptoms, but how could you tell the diagnosis apart? And that was a tricky thing, and BNP was emerging at that time as a potential solution to the problem. It's now in the guidelines. This is from the European Society of Cardiology. It's their guidelines, and you can see here that BNP, NT-proBNP, it's a class 1 level of evidence. It's a class 1 recommendation with a level B of evidence, so it's fairly well ingrained in the guidelines, but it wasn't at the time, and there were some issues that we were wanting to solve. And you had to use an animal model because you just couldn't do certain things to humans, not even medical students. It was unethical. And so some of the questions that we had was, how much does the BNP biochemistry form change from when it's released from the heart into the peripheral circulation? And as heart failure develops, does the production and the processing change? And what are the forms that are relevant to the sheep model, because you need to know exactly how your model's performing, and are they cleared in the same way? And all of those things are important, because if you're going to design a test or measure something in the blood and it's going to be useful in your model, all of the clinicians, you want to make sure you're not missing something. So these are the very clever folk who live on the 3rd floor. They don't look like trouble at all. And they have their very well characterised model of heart failure, where they basically stick pacing wires in the heart and they pace it at set rates for a certain period of time, and the sheep develop all the characteristics of hemodynamic and neurohumoral, that is hormonal heart failure, and it's an excellent model, and they're a highly skilled, world-renowned team for doing this. So the first thing, I'm going to bore you now with a little bit of biochemistry. The first thing we did was create the assay, and it seemed to perform very well, and you could measure off here, this is the standard curve, and you could measure off here what your achieved level in the sample was. So the first thing we're trying to figure out, do the forms differ from the heart and the circulation? Is it cleaved in some way? And these profiles here are just size exclusion chromatography, which if you see this peak here, which looks like that, and if it's at this end over here, it means it's a bigger molecular weight than it would be if it eluded down here. And each of the peaks in each of these profiles showed that the gross overall biochemistry was the same. There weren't major changes between the heart and the periphery, unlike ANP, which is down here, and you can see it's got a bit more of a complex profile. So that was the first part of the question. On a gross assessment, the ovine BMP forms didn't change much between the heart and the peripheral circulation. What happens when you ramp up heart failure? And so you ramp up heart failure here by increasing the heart rate, and you can see here as it increases from 120 through to 225 beats per minute. What happens is your left atrial pressure, so the pressure inside the left atrium in the sheep's heart was going up, the left ventricular pressures were going down, mean arterial pressures changing, and the cardiac output went down as well. And if you altered the heart rate, the animal would respond accordingly, and you could have beautiful changes. And so this was a fantastic model for this, and we knew exactly what was going on in terms of the model and its induction. And to cut a long story short, again, more chromatography, size exclusion up here, and this is a different form of it called ion exchange chromatography, where you can tease out what's underlying under here. And the only difference really was this peak here, this one here called number 4. And this only appeared in the sheep who had severe heart failure, and in the normal sheep it was just the same here. So as a general rule, there was a slight change in the BM forms that were measured across heart failure, but the assay still detected them all, so that was a good thing. Then we came to the next problem, which was BMP and NT-proBNP. So BMP is produced as a parent, and then it's cut in half, and you have BMP and what's called NT-proBNP coming from that. And then we had to figure out how NT-proBNP performed in that particular model as well. And to cut a long story short, they have very different concentration profiles. So the chromatography here is a better way of looking at it. And so you can see these peaks here. This is the NT-proBNP and the BMP here. They're about the same size and the same size here. But when you put the sheep into heart failure, the NT-proBNP vastly outweighs the BMP. And so this was an interesting point, in that both BMP and NT-proBNP were relevant to the sheep heart failure model, but NT-proBNP was lasting a lot longer. And the question is, why is it doing that? So we went on to the next thing, which is, are they cleared, are they produced, or are they cleared at different rates? And the shorter answer is we used a mathematical technique for the mathematicians in the room, wherever he is, there he is, called deconvolution, which basically is a fancy way of saying we know what you're doing. And the whole basis behind that is we would pace the sheep and it would induce an increase in both of them, but the BMP would drop away very quickly, but the NT-proBNP would take a lot longer. And using deconvolution, by subtracting, because they're produced from the same parent, by subtracting the BMP, which we know the half-life, you then take that away from what the NT-proBNP, and this is the delay curve here that we can see. The BMP and the AMP came down very quickly, but the NT-proBNP took a lot longer, and it ended up being about 69 minutes versus about 4 or 5, maybe 8 minutes in the sheep here for BMP. So it had quite a much longer half-life. And this is reflected here as also if you block the clearance pathway for BMP, so you can stop its cleavage or you can stop it being removed from the circulation. You see the BMP levels here in the normal animals, they're all around the 60s. As soon as you start blocking it, it goes up and it ends up at 168, whereas NT-proBNP is trucking along and is quite happy. So that was the first evidence that they're cleared from quite different pathways. So this was all very good and got written up in a book, and that was great. But it's not over. So this is the latest algorithm from the European Society of Cardiology here, and you can see here, a patient comes in, they're breathless, you do all the clinical thing, and the first thing you order, that's the recommendation, NT-proBNP or BNP, they're both used. But all of that biochemistry stuff I've just told you about, it doesn't end there, because the genetics of BNP, so it's produced in the nucleus, it comes out, it's cleaved, one's biologically active, one's not biologically active, it's got sugar groups, it's glycosylated on the NT-proBNP, it's got all these other different things. And people in our group, Linley and Alamander, are looking at this because this actually has implications, particularly potentially the glycosylation, because big things impact on the diagnostic performance of BNP, such as obesity, atrial fibrillation, things like that. And there may be ethnic differences which are reflected in the amount of glycosylation which is in here, and this is important. So things never end, they always carry on. So that was the second lesson that was learnt. You come out and you've got a newly minted PhD and you're thinking, oh, I'm ready to take on the world. But then you realise that an essay is submitted or a thesis is submitted, but it's never finished. Yours is just one thing. Now, to those who are writing up a thesis, don't get depressed. That's not what that means. Your thesis is important, but carry on. So then we went off to Finland, and Finland's a beautiful country. It's got the lovely northern lights there that we took a photo of, but one of the drawbacks of that was that you end up sending Christmas cards and things like that with the real Santa. And we were all very excited about this. This chap here was very excited. She was a little bit nervous. He wasn't interested at all. But they have this really amazing thing you can do there, and it's called the wife carrying competition. Now, it just has to be a wife. It doesn't have to be your wife. It could be anybody's wife. And it was suggested that perhaps we could partake of this. The look I got back, no way. And she was right. But the big attraction of that is you win your combined weight in beer. And that was the big attraction. But anyway, all we could do was observe. The reason I went to Finland was to learn how to run this. And this is the isolated heart. And so this is a rat heart. And you take it out of the animal, obviously, and put a cannula in it. And then you can... oops, can I do that again? Is it going to work? No, it won't. Anyway, you saw it beating. That's good. And then you cannulate the left ventricle so you can get pressures, and you can pace it here. But you can also measure the coronary artery, what's happening, the constriction or relaxation of the coronary arteries. And it works very well just through measurement up here with these cannula and the pressure here. And so you can use it to measure what's happening to the relaxation of, or the constriction of the coronary arteries in the heart. And that's what this data here is. We published on the hormone called ghrelin, which comes from the gut. It's a gut hormone, but it has quite a prominent cardiac activity. And if you give it at the right dose and dose responsive, it increases the constriction in the arteries over a period of time. Or you can use it to measure contractility. And this is from Jaakko Pihila, who was in the Finnish lab, and he came back to Christchurch and did a postdoc with us as well. And this is looking at EPO, erythropoietin, which at the time was undergoing kind of a bit of interest as a cardioprotective. Unfortunately, it fell out in the clinical trials, but you could measure the improvements there that it did with contractility, such as developed pressure there. It would increase it, and then it would improve it in the reperfusion phase of ischemia reperfusion. So that was what the model was all learnt about. Then came back to Christchurch and was reading a textbook, and we all know what textbooks are like. They are the Bible, they are the Word, and you have to learn all this, and they're kind of like an old school teacher who says that I've been here for 30 years, I've always said that, and I'm not going to change. And I was looking at this one day and reading it, and I was looking at the paper from Nature that I had in front of me, and I thought, nah, I don't believe that textbook, I'm going with the paper from Nature. And it concerned this thing. called a signal peptide, and a brief biochemical refresher here, or new for people who don't know it. So when you're making a protein in your body, you have the ribosome and your RNA, and it gets together with a thing called the signal recognition particle and GTP, and they go, hey, that's cool, let's hang. And they all form a complex, and it goes into this thing called the endoplasmic reticulum. And this little bit just here, is called the signal peptide, and it's the very first bit that gets made. And that bit then gets chucked away, and that's what the textbook was saying. It gets, once it's done that, it's given all the information where the protein's meant to go, and that's all it does. And the textbook was saying this, and I was reading the paper, and it kind of didn't. The whole thing didn't gel. And this is how science works. You know, things get pushed along. And that whole hypothesis came from this guy here, Gunther Blobel, who got the Nobel Prize for it in the 1970s for working all this out. So we went back to our favourite molecule, BMP, and it has a 26 amino acid long signal peptide, and we thought, let's make an assay to it, which we did. And so Mithri Siriwardena did a lot of this work. She was a clinical cardiologist who spent a lot of time walking the wards in the hospital in the middle of the night getting blood off these people who'd had heart attacks. Quite amazing, the tenacity with which she did this. And so we set up an assay and measured a whole bunch of data and samples, and this is from work from Swaitonia and Mark Richards, who did an elegant study called the gradient study, where they put cannulas into people who are in the intensivist and the clinical cath lab anyway. They were there because they were clinically indicated to be there. And while they had them there, Swaitonia and Mark would take blood samples from various venous beds, draining organs, and also the arterial flow as well. And this confirmed what we already knew, that BMP or NT-proBNP was primarily coming from the heart. That's what this peak here means. And the signal peptide of it was also coming from the heart, but it was coming from a whole bunch of other places as well. And this caused a bit of scratching because we thought it was exclusively a cardiac hormone. So we went to Dunedin to the very clever Torsten Kleffman down there, who was a mass spec expert of renown, and ran the facility down there. And so we purified out what our assay was measuring and what we thought was BMP. And first thing he said to me was, never waste pure thoughts on an impure protein, which is very good advice. So we made sure we had the preparation very pure. And then we ran it through a reverse phase HPLC column and got all these different peaks. And then he went and looked at all the different peaks, and it was there, but it had all these modifications on it. And so it was 142 Daltons bigger than it should have been, or 102 or 128. And he said to me, this is weird, I've not seen this before. And he went and looked in the literature, and he said, actually, these things are real, and I'm convinced that you're actually measuring what you're measuring. But this is a bit of a big deal, because this is the first time I've seen this on something like this kind of protein. So I went, okay, that's great. So we published that, but the big thing with BMP signal peptide at the time was that in patients who were coming in with a heart attack, the markers at the time called troponin and CKMB, they took a few hours to come to their peak, and they would rise, but they weren't really at their peak until, say, 8 to 12 hours after the event. BMP signal peptide rose very quickly. It was up and then came back down. So by the time these things were peaking, BMP signal peptide was basically done and gone. And then BMP and NT-proBNP would rise later as well. And it would only do it in these patients who were having an MI, in this case, a STEMI. And so this was very good. And this is where Siri was doing a lot. Now, Siri hates having a photo taken, and she hated having a photo taken so much, even though she's now in Australia. And here, I dug this out, this is the ABC News in Queensland 2018, and they've basically saved a woman's life, and this was a big deal that made the news, and she's still trying to hide behind a piece of equipment here to get out of the photo. So if Siri watches this online, I've got you now on a photo, and there you are. The next bit of work that we did was patients who had suspected coronary artery disease, and then we wanted to know, well, how quickly can this thing be turned on and off, BMP signal peptide that is. The answer is very quickly. And so Siri took great delight in subjecting actual clinical patients, but then also members of the CHI to what's called a dobutamine stress echo, which is where you lie on a table, you have a whole bunch of lines in you, and you're very prone. And she gives you this drug called dobutamine. It makes your heart pump faster, and then she gives you a bit of atropine just to top it off. And it's quite a soporific experience. It's otherworldly. But what we found was that BNP signal peptide went up very quickly, but other known cardiac markers such as troponin, NT-proBNP didn't really work. But this was so we confirmed that BNP signal peptide could go up and down very quickly. But what was really interesting to us was that the high sensitivity troponin T would go up even in normal people, which is in the blue here, and they would go above the 99th percentile of what was considered normal. And so when we first looked at this, Mark commented to me, Mark Richards said, have we been giving people heart attacks all this time that we've been doing? And the answer is no, and it's a bit more complex. But that confirmed that BNP signal peptide was actually an inducible marker of ischemia. The next thing we thought, well, okay, it's coming out, we can measure it. Does it do anything? And we did a lot of work here in isolated rat hearts and in sheep. And so we presented this at the European Congress of Cardiology in 2018. And so we did the isolated heart model where we would give it as a potential therapeutic before a period of 40 minutes ischemia or at reperfusion only. And this is really more clinically relevant because you don't give someone a drug before they're going to have a heart attack. You're usually doing it afterwards. And to cut a long story short, in the isolated heart, the troponin reduction was massive. This was beforehand, it would drop, and then it would be released afterwards. Believe it or not, these cells actually here are, you can actually measure them and count the numbers, which was a fun job. But there was a reduction in tunnel positive staining, which means that there were less cells that were being killed when we gave the drug. And it was doing this through the phospho-ERK pathway, and it was almost doing it exclusively through phosphorylated ERK1. So you can see here the blue bars get increased as we're giving an increased dose of BNP signal peptide, and phospho-ERK2, nothing was really happening. At the same time, there's an intracellular protein called AKT, and phosphorylated AKT goes up, it automatically goes up. It's a natural defense mechanism that your heart cells have when they undergo a period of ischemia. And BNP signal peptide, at the same dose as it was turning ERK1 on, wasn't doing anything to AKT. When you went to a higher dose, it would reduce the AKT. And this actually fitted with a lot of other observations we had. If you give too much of it, it actually turns into a deleterious agent, which is consistent with a U-shaped curve that you see in therapies and drugs. So then we went from the isolated heart to the sheep, because we've got a lot of them, and we've got a very good model. And Chris and Miriam and others worked together, and they made a ligation model of myocardial infarction. It was a snare, and so they would ligate off the artery for a period of time in the sheep, and then you would have an infarction. And that's just a picture there of the surgical workup. And so we did a number of animals. And the interesting thing here was that it didn't really do anything to the hemodynamics as it was going on, and it didn't really do anything on echocardiography except maybe a 10% increase in the ejection fraction of the hearts that received BNP signal peptide afterwards. But then, in terms of the troponin, we got a similar result to what was happening in the isolated heart. So these are the achieved levels of what we were infusing into the animal. You can see it's all over the place, and then it drops away very quickly. Yet despite all that, we got this massive reduction in what was released in the troponin from the hearts. And then the actual area of the heart that was affected was the final size of the infarct in the heart was reduced as well. It's the red versus the blue here. And the tunnel stain controlling again, the number of cells that were being programmed to die was actually reduced. And then the caspase 3 staining, there was a tendency for that to be reduced. And caspase 3 is a marker of programmed cell death in this particular situation. So this was all very good, and I just want to acknowledge all the people here who took part in that study because it was quite a lot of work. So we've got Siri there as well, and Jackie Cannon, who was helping with the caspase 3, Priska, who did a lot of the Western blotting, and Chris and Matt, who were involved with Siri when doing the sheep work. So at the end of the day, we'd shown that not only does this thing circulate, but it actually has biological activity as well, and it does it through the AKT. and the ERC pathways, which is why it got accepted into the European Congress of Cardiology for this particular talk, which was quite good and a fair reflection of all the hard work of everybody who'd been involved with it. While all that was going on, we had a number of different assays to different signal peptides being made. So we had all the natriuretic peptides sorted out, ghrelin, that hunger hormone I talked about before, insulin and EPO, and we had a number of publications that came out, and we had all this stuff, and we didn't want it to end in this way. So you do all this work over a number of years and you just get one thing and everybody goes, well, is that it? And at this point, we had discussions with Otago Innovation in Dunedin, and we set up this company called Upstream Medical Technologies. I originally just wanted to call it Upstream, but the amount of arguments, it's just like... Anyway, I relented and we called it Upstream Medical Technologies. And this is the website, this is the updated website, which is about to go live, hopefully in the next month or two. Our current website is a bit naff, really. But that's what we do. And it was set up through Otago Innovation. And I have to say, David Christensen and others from Otago Innovation and Colin Dawson, who was there at the time, they were very supportive and very keen to push this through, and it happened. And this is the Extreme Upstream team. So we're led by Kieran Gina, he's the CEO, who is also a partner in Pacific Channel, who are our major funders at the moment. Jan Powell, who works at ECR, but she had a lot of time at a company called Shire in America, which is a big advice kind of consulting company and funders. And Jackie Lee from the lab, she's our CTO, we second her a bit. Jessica, who is our scientist, and we want to give her a full-time job if we can do it. These are our advisory board. So we've got Frank Peacock from Houston, James Januzzi from Harvard, Mark Richards, of course, Martin Tan, who's an ED advisor, and Martin, if you're watching, we really want to make you CMO, so please call me. Louise Cullen, who's a colleague of Martin Tan in the ED department, and John Pickering, who is the Scry Master. There's that word. Consultant, and he does a lot of our excellent statistical analyses. There's Garth Hendry. He's actually a graduate from the University of Otago and PhD in biochemistry, and he decided one day that sitting down a lot and writing patents on other people's work was much more interesting, and he's actually doing very well at it now. He's an excellent patent lawyer. He lives in England. So this is the entire team we have working with us as well. David Christensen from Oil is on the board, along with Roland and Colin, who are also part of Pacific Channel. So what do we do? And this is just an indication of what happens when you set up a company and you think you have an idea and you think it's going to go okay. We've got 40 issued patents, and we've got one which we're hoping to get through. That's to be confirmed, but if we can get that into the company, it'll be really great. 2 of those patents are actively leading now in what we're actually doing and pursuing and getting further fundraising. So that's just an indication. You can get a lot of patents, but the hit rate doesn't necessarily transfer into a product. This is from the European Society of Cardiology, and this is a sort of a schematic of what's called chronic coronary syndromes. So people go through life and they have what's called a subclinical phase where, by virtue of just getting old, your cardiovascular risk increases. We all know that. Then you might have some kind of symptoms or something like that. Then you might actually have an event. And the whole point of therapy and monitoring of people is to take them away from this high-risk red area here and get them down into the green area where their risk is low. And the whole point of what we're trying to do is to provide monitoring tools so that we can take people from here, if they're there, and get them down to there. That sounded quite good, didn't it? Anyway, so we've got 2 studies that are going on, and this is what's called a receiver operating curve here. And for the sake of brevity, you want a line that goes up here to this left-hand corner and goes straight across. And that's what's called an area under the curve of 1. If you achieve that, you have achieved a diagnostic bliss. You have the perfect test, and you can drop the mic and go home. You'll never get better than that. If you have a test that goes along this orange line, it's like flipping a coin, and you really should start looking for another job. And what we've got here, we've done 1,900 patients from a space study here in Christchurch and a study called Fast Track, which is a multi-centre study in the US. And what we've done is we've improved this green line. This doesn't look like much, This red line to green line difference here doesn't look like much, but it actually means everything. It's an improvement of around about 17% in specificity, and I'll come to that in a bit more later. So that's for the diagnosis of unstable angina in the ED. What about the people who come to the ED, they're not having a heart attack, but the doctor's still suspicious about whether they may present within the next 12 months? Again, our marker C this is the regression. It's a significant regression. It adds to troponin and a history of coronary artery disease, or whether they're going to come back again. We're inching this line across here, this orange curve, we're inching it over here to the 1. And only when you add those two, our markers and troponin together, are you getting a significant improvement in that predictive ability. And it also correlates with people who have a history of coronary artery disease as well, which fits in. So if you include troponin, our marker C, a patient's risk of cholesterol and their age, you can have a good significant predictive ability of whether they're going to have coronary artery disease or not. So that's all interesting. This is the latest one that we're trying to get into the company, and hopefully we can. We'll see how it goes. And what this does is troponin T is one of the gold standard biomarkers for the diagnosis of MI. This thing called marker D, you realise I'm calling the marker C marker D because if I did, I wouldn't have a job. This is what business does to you. You're allowed to be vague. Those who are doing the work know what it is, but you know, it's just one of those things. This particular graph here, we took this to Barcelona 2-3 weeks ago at a conference, and I presented this to the people from Roche Diagnostics, that our marker D was actually improving the rule out here. So we're improving the predictive probability of someone who doesn't have a coronary event when the HSTNT is very low. We're improving it from 89% to 95%. I showed him this and he literally thumped the table and got up out of his chair and he goes, I've never seen anything do this to troponin T before. So we found that quite encouraging. So hopefully, if anyone from Roche is watching, call me, you know, that's good. And then this is the final, all of that kind of stuff works into this. And this has been worked out by the very, very clever John Pickering. And this is because this is his bread and butter. This is what he does, clinical pathways in the hospital. If you come in, this is based on our 1,900 patients, if you come in, you get everything done to you at 0 hours, and a group of people are going to be sent off because they're definitely having an MI. And then you can get graded into the next category of unstable angina, which is one of the next ACS conditions you're worried about, as to whether they're having UA, and you've got low risk, intermediate risk, and then high risk. If you then take the delta of our marker between 0 and 2 hours, you end up being able to boot out 19% more patients who are at low risk and can go somewhere else, and you're capturing another 2/3 more of people who are at high risk with a PPV of 25.6%, which means 1 in 4 of them is actually going to definitely be an unstable angina. And when we put this in front of our consulting board and other cardiologists in Barcelona, they thought this is actually pretty good, particularly taken with the PPV there, they thought that was quite good. So this is all positive stuff going forward. And we're hoping to carry on. But I'd just like to share with you experiences of getting money out of private equity and venture. It's not like getting money out of a grant. When you write a grant, although the methods are the same, it's a business plan in a way, getting money out of venture capital people is a totally different beast. When you go to the HRC or the MB, even if they don't give you any money, they know that you know that they've got money. It's a different story here. So these are the top 5 repeated replies that have come back asking VC funds and private funders the most important rule for success. The first thing is, how did you get this number? The next thing is, please stop, don't ever call again. Then they say, what, get out of my driveway. Then it's, my children can see you, for the love of God, please leave. And it usually finishes with, yes, that's him, officer. There you go. So these people are very, very close-chested, and you have to, in the commercial world, it's all about relationships. And there's a phrase that they use, it's called the quality of the money. And that doesn't mean just the money, it means, do they believe in you? I mean, it's personal, which is a bit scary, isn't it? Anyway, but once you break those barriers and they believe in what's going on, they will back you to the hilt, and they will follow up with money. It's a really interesting roller coaster ride. So, next we're getting to COVID. Everybody's sick of COVID, from the full-on frontal lobotomy that you would get here, from that through to staying at home, day 1 to 5, everybody knows what that difference looks like. And just some words of wisdom from Nigel Molesworth again. History started badly and it's been getting steadily worse, and I think we can all relate to that in some way. So I was sitting at those of you who heard the story before, I'm sorry, but I'm going to say it again for those who haven't. I was sitting at the dinner table and I got a call from Richard Blakey at home because we were locked down. And he said, MB's putting out money, you should try and get some of it. And I went, okay. And I wrote an email and I looked at it and I thought, this is great. And I wrote a expression of interest. 70 minutes later, I got an email back from MB saying, this is great, apply. 1 month later, they gave us $1 million. Now, if anybody from MB is watching, please repeat that over and over again. It is a very efficient way of doling them out. It was an unreal experience. I don't think we'll ever see anything like that again. So we got money from MB to develop WAT, which are antibody assays, to measure antibodies in people in response to COVID. Everybody knows what antibodies, vaccines, the whole 9 yards there. So we raised them in rabbits and goats, and then we set up our own assays. And these were developing our tests in the recruiting patients via the Nichols Centre, and that was the whole shebang. In terms of making the tests, so we make the assays to measure the COVID antibodies. We want to measure the spike protein as well, which is coming from not just the vaccine but the virus as well. And then to apply existing clinical tests and novel ones to see how they perform. We wanted to bleed up to 3,000 people and collaborate with anyone and everyone. Our tests were being performed on what's called the Luminex machine here, of which Sarah is the queen. And we were benchmarking those against the Roche machine. So this is a very long-winded way of showing what happens across Pfizer vaccination. So it's comparing our assays and the Roche assay against people who've been vaccinated. And New Zealand was a really unique place at that time because we didn't have COVID, but we had the vaccine. So everybody was getting vaccinated. So we were kind of like this really amazing, unique incubator. And what happened here was that this is our Luminex assays, and you would have antibodies created to the spike protein as well as to the receptor binding domain of the virus, and that these benchmark very well against the Roche Cobas assay, where you would see they set everything to 0 because with blessed German efficiency, if you did not have the condition, your value must be 0. They've never heard of this thing called interference. But you would get these beautiful curves. But our assays were working just as well. We then measured about 9, we've actually measured 11, but this is 9 different markers across the period of vaccination. I still don't think this has been published, which is why we're putting this together. And basically what this graph shows is that for NT-proBNP, TNT, a whole bunch of different markers that are used in immunology, hematology, inflammation studies and everything, getting vaccinated across before first vaccination 1, your second vaccination, and then 2 weeks after your second vaccination, nothing really changed. Everything pretty much stayed the same, apart from GDF15, which in about 60% of individuals would show some kind of differential response. It wasn't major, but it would change a little bit. This actually fits with what's coming out in the literature. GDF15, it's everywhere. It's used in cardiovascular medicine, but it's actually quite a responsive marker in COVID biology. So it's actually going to, it's probably going to be used for a long time coming in that setting. The other thing was, could we figure out, based on what your baseline levels of certain markers were, what was going to happen to your vaccination? And it turns out that if you combine your fibrinogen level and SUPAR, which has been studied in our lab by Janice Chu-Harris, your baseline level would predict, so if the higher the combination of those 2 markers, your 4-month Roche antibody level would be low. But if the lower they were, the higher, so there's an inverse relationship going on there. So it's very small numbers, but that was interesting. The other thing that interested people was vitamin D, because everybody was running around and saying, hey, vitamin D, vitamin C, do all this, you won't get COVID. Well, no, that's not how it works. Vitamin C and vitamin D may assist your immune system, but they're not going to give you immunity. That's very clear. And what this showed was that there was a trend with your previous vitamin D level here at the pre-vaccination you would tend to have a higher response of your antibody levels to the vaccination measured by the COBAS assay. We use this assay because it's an indicated one. And your vitamin, it's a slight positive relationship there with your vitamin D levels. It's not significant, it's interesting, and that's really about it. So, and the assays that Sarah has created are very, very good. And I just want to make a shout out here for IgA. I think IgA has been neglected far too much, because if you look at the area under the curves here, the clinical assays from Roche and the COBAS to the RBD and also to the nucleocapsid protein are pretty good. And our own IgG assay is working good, but IgA here is up there. And I'd be really interested to know from immunologists and infectious disease people, it just seems IgA seems to be the unsung hero in all this and warrants further inspection. Now, how long do antibodies last for? Again, we could do this because the vaccine from Pfizer only gives you antibodies to the spike protein. So we got 23 people who've been vaccinated and no COVID, and then 30 people post-COVID in through the study, and we watched. So here, this is the spike protein, and you can see this is the 2-week and then 4-month after vaccination. There's a decay, but they've got measurable spike protein antibodies. And then out for a period of time, they're still there. Some of these people have been revaccinated after COVID infection. For the nucleocapsid protein, as expected, vaccination here gives you no nucleocapsid antibodies, and so you can use this to figure out who's been vaccinated and not had COVID and vice versa. And it turns out that out to 2 years afterwards, there's still measurable antibody responses. So your memory cells in your immune system, your B memory cells that are floating around, are still tuning out this stuff even after 2 years. What that means, we don't know, but it's an interesting fact. And so on that particular study, well, I really want to thank Scott Loeffler and Aaron. It's really hard to get a picture of Aaron. Has he done something? Is he... but the COVID Queens here, Lorraine, Judy, Sarah and Jackie, always ready to fight the good fight. Steve Chambers, the godfather of infectious diseases here on campus, he was being extremely helpful. Barry and Judy in haematology, doing T-cell work, absolutely fantastic. And John O'Donnell, clinical immunologist, and Paula Keating from the CDHB as well. Absolutely fantastic collaboration, very, very much appreciative of it, and hopefully we can continue it in the future. So finally, I just want to thank a few people who have been quite important along the way. Obviously my parents for conceiving of the idea of conceiving me. That's fantastic, and for teaching me things that are all important about life and what to do, and for introducing me to the greatest valley on the face of the planet. There will be no discussion entered into here. This is the Waitaki Valley, and this is facing west up to Aoraki, just taken from our family batch. And we had many a good time. And this is actually a photo of my parents on a motorbike that had just been bought by the farmer on whose land our batch is, and he came along and Dad tapped Mum on the shoulder and they stole it. And they went into Kurau and didn't come back for an hour because they went and had a cup of tea. So that was the kind of thing that we got up to. My brother Nick as well, were very supportive, and these are my two brothers, Steve and Phil. This is at Phil's graduation in Cambridge, and this is one of the I'm so glad we're in New Zealand, this is one of the peculiarities of English sort of upper brick universities. Both of them graduated from Cambridge, and Steve was a fellow, and this is Phil's graduation, and this person here had just come across to Stephen and told him to get off the grass. And Phil's pointing this out, and Stephen's smiling, because Stephen said, I'm a fellow, I can walk wherever I like. And she went, oh, and left. Only in England, right? All about the grass, which is ironic, because they did caterpillars at the start, so you know. Anyway, and then there's love, and I wouldn't do it with anyone else, but you did push me off the stairs there, so you can get the photo to your whole self. And then these guys, two of them like to play dress up, and this was at her, was it admission to the bar? Yeah, he'd already been to the bar, and you can tell by his drunken look. But anyway, they're in London now. This guy's in Wellington, finishing up his, well, doing a very good job of trying not to finish his BCom, but he's getting there. This one's in the audience there, cutting hair and making sick beats. Yeah. And then this guy, he's a lot of trouble, and unfortunately the apple doesn't fall far from the tree, but he's a good kid. And then also to colleagues, hoa mahi. I couldn't find a better group to work with, absolutely fantastic, particularly these guys here. No, they're not halos, all right? But Matt, Lou, Sarah, Noel, Liz, Joe, Janice, Tim, Sarah, I've done her, Andrew, Mark, who's doing an MB, CHB and a PhD at the same time. Why? Anyway, Sarah, Linley, and then Jackie, Joy, Kirsten, who's just come on board. Good on you, Kirsten. Jay, a PhD student, and Taylor Keys, who's done a B.Med.Sci honours with us while he's lifting weights or something there. Absolutely fantastic team. I'm very privileged to kind of be standing here on the back of all of the hard work that you guys have done. And I would particularly want to thank these kairahi, or mentors in particular, Tim and Eric, for believing me in the start and giving me the introduction to the PhD. And I'd like to thank Eric in particular for when I was doing my PhD, you also wrote my PhD in pen on the other side of the printed page, and I could not believe that I would give you 20 pages and I would get 20 pages of writing back. Absolutely amazing, absolutely amazing. Also, Prof Nichols, everybody's got a Gary story and we've named our centre after him, but your encouragement and support along the way has been absolutely fantastic. Mark. I'll put this photo up here because he's getting another award. How many awards can a guy get? But he's got his suitable optimism there. You know, not content with running the CHI, he disappears off to another country and builds another research group there. I think a lot of us here realise the contribution that Mark has made and the support he's given. Absolutely fantastic. So if you're watching this online, Mark, your support and encouragement over the years has meant an awful lot. Martin Tan, from the ED department, he's a great guy, Martin Tan. I really want to thank him. No, he's not here. Really want to thank him for letting us loose and being able to walk around the ED with him and actually watch what goes on. The amount of learning that happens is fantastic. And then the girl from Balmain. I can't count the number of times that studies and ethics proposals and just general conversations and making things happen. And now I know you won the Bake Off Award there, but Lorraine, you're an absolute treasure and you've done pretty good for the young girl from Balmain, I think. And it's been a real honour and privilege to have worked with you. So, finally, it was recently suggested via a send or email, is she here? She better be here, that you are what you wear and that you can always change careers if it doesn't work out. Maybe to something like that. Now, you come to work in a blue top and all hell breaks loose. And I thought this was highly unfair, and so I think it needs to be zhuzhed up a little. So we need Chris, what's Miriam done now? Nick, yellow had nothing to do with it, but she took the photo, and the instigator, Miriam, it wasn't me. And I'll just say that, you know, all of that's been put together by Revenge as a Dish Best Served Cold at OPL's me. So on that wiggly note, I'd like to thank you all for listening, and the Kai is almost here. Thank you very much.
I'm Trish Priest, the acting Pro-Vice Chancellor of Health Sciences at the moment. I think I last was in a lecture paper in this, what was in clinical school in about 1985 when I was a 50-year-old student. So it's great to be back. I have been given the task of thanking Professor Pemberton for the lecture and making comments about the content and significance of the lecture, which is a bit of a big ask with such a big lecture covering so many things and such an interesting career. But I'm going to give it a go. So we were promised caterpillars, coronaries, and cardiovascular disease, and we've certainly had that, and a career spent testing boundaries. So as I listened, I was trying to think about themes that were coming out of Chris's talk, and these are the things that I came up with. So I won't reiterate the content, because there's a lot of it, but I think the themes that I identified were kind of the epitome of science, really. So curiosity, which has obviously driven Chris into science and lots of different parts of science, and that thing where you see something and think, oh, what's going on there, and then follow it up. That's really fantastic. Mentorship and teamwork is obviously a really big part of how we do science, and has been a big part of your doing science, and it's really fantastic to see you acknowledging all the people who you've worked with and the impact of them on you. Obviously, BMP and various associated peptides is a theme, and I think the way that you've spanned from the biochemistry and some of the stuff I haven't really understand, and across through to thinking about diagnosis and prognosis and potentially treatment is just a brilliant illustration of translational science and what we do really well at the University of Otago in our medical school, where we have all of the parts of that spectrum, and you've kind of got in there in all the different areas. The commercialisation and sitting out your company and trying to interact with a whole other world of finance and so on was really interesting, something that I have never tried to do, and that sounded wildly tempting. But good on you, and it's really fantastic, the progress you're making, and the potential for coming up with ways of diagnosing people's prognosis early so that we can reduce their risk, which is obviously important. And I guess finally, in terms of context, it's just worth noting that cardiovascular disease is one of the biggest causes of death and loss of quality of life in New Zealand, as in many countries. So this is a really important area to be doing all those cool things in. So thank you very much for explaining all that to us, and congratulations. And I think I hand back over to Suze.
Kia Chris. I think not only along with being curious, I'm really grateful for you having so much courage in your career, and I really picked that up. Both when you started with the longest mini I've ever seen in my first language interview speaking do, and with such boldness, but I think that really was a beautiful tuhu actually for all the courage that you've had in your career. And I know you're far more proud of you, but I hope you also know that the UOC team is really proud of you. We're really grateful to have you as a taonga amongst us, and actually look forward to your new adventures and what else you will do now that we have to address you as Professor Pemberton. Which sounds, although you didn't go to Cambridge, I think it sounds very, very posh. So, I'm now going to invite Professor David Murdoch as our Vice Chancellor to lead the academic party, and would like to ask the Pemberton whānau of Professor Pemberton to join the academic procession, and then we will all be able to go and talk and have a quiet conversation and tell us how great it is out on the floor. So thank you, David.